So I have to modify the topic a little bit and the number of slides that I will be presenting today. So, so I've tried to follow the guideline as sent by Dr. Resberg. So I will talk about research, we do the tools and our research group and the institution, justification for the use of the HPC and how our research depends on this HPC and some research results. And the research result will be, I would say, a snapshot of the kind of thing we do, which I will present result on iron at extreme pressure and at temperature conditions and oxygen defect in metal oxides if time allows, and metal oxide alloys. And I will talk about the computing challenges and successes we've had using the HPC. So talking about the research, so we basically focus on metal oxide alloys. We have a number of collaborators that are spread across the world. So we have a collaborator at Lehigh University, United States. And basically we have expertise which we combine to achieve our research objectives. So for the metal oxide alloys, one of our colleagues, Professor Kumar, deals with DFT, SPR, KKR methods of material modeling. I'm basically DFT person and the SPR, KKR as well. We have our colleague from Marian Uwambi University in Congo. Then, of course, at UNISA, and of course, at Czech Republic. So this kind of material we consider. We are quite flexible. So metal oxide alloys, transition metal oxide, bulk and surfaces, two-dimensional system like graphene, carbon nitride. And of course, of recent, we start to look into spectroscopy like Raman, like angular resolve photo emission spectroscopy, whether experimental and theoretical. So the tools we use our main tool are HPC, of course. The high performance computing at HPC is the main thing we use. We use the quantum espresso code, highly parallelized for material modeling. Some of us use Gaussian, exciting. We have AT-AT for preparation of metal alloys, basically to prepare alloy system. And we have Material Studio, and of recent we are using Schrodinger as well. So we have this suite of codes that we use depending on whichever one is convenient for the students. So, so why do we use HPC? So most of the codes we use they are basically solve Schrodinger equation. And so these codes solve Schrodinger equation cast in many electron system, and the equation is usually framed this way. And because a material contains of infinite number of electrons and atoms, so if you have to solve this kind of equation, which has to be done in a very self-consistent manner, so it is not possible to do that on the normal CPU. It has to be done on the HPC because it is data intensive and it takes days, sometimes weeks. So this is not feasible on CPU, so we use the HPC at HPC, and pretty much any of the first principle codes rely on this kind of solve this kind of equation, whether you are talking about quantum espresso or Material Studio or VASP. They basically solve this kind of equation, and so we use clusters, and our codes are highly parallelized. Many of them we get online. They are open source, quite available free of charge, except few ones like Material Studio, VASP, and the rest, which most of the time we don't normally use anyway. So so let me focus on the S3 absorption as one of the techniques that we have been used. So basically, let me just introduce you briefly to S3 absorption techniques. So basically, this technique provides bonding, information at bonding and structural information, and is sensitive to perturbation. So therefore, if you have a material system that is very sensitive to, if you create some kind of perturbation in your material system, so our proposition is that you can use the S3 absorption to kind of fingerprint the kind of perturbation you have introduced in your system. When we talk about perturbation, we refer to things like maybe defect, for instance, or any structural inhomogeneity you have introduced into your system. Extra absorption probe unoccupied states. That is very peculiar to the absorbing atom. And so it looks like this. So basically, you excite atom deep in the core into the continuum above the Fermi level. And when you do that, say you obtain this kind of spectrum. Usually, this is a kind of, okay, so if you excite atom from the S core orbital, for instance, to the continuum, then you have what you call the KH. If it's from the 2S to the continuum, you have L1H. From 2P to continuum, you have the MH. And basically, the kind of spectra you obtain looks pretty much like this in most cases. And you can identify three regions. You have the pre-H region in the spectrum, you have the H region, and then you have what you call the continuum. So basically, an H results when a core absorbs electrons greater than the binding energy of the orbital, and the electrons are excited to the continuum. So extra absorption is element specific. So this means that you can actually focus your beam on a particular element to probe the electronic properties of that particular atom. So that is why it is quite useful when you want to probe things that happen in maybe surface catalysis, for instance, or the local environment of an atom. So it's quite amenable for that. So, this thing is caught. Anyway, computation like this is how we do it. I won't like to bother you with the details. So basically, we obtain the cross section and of the spectra, and so this. So what is important to know at this point is that some of the features of the extra absorptions can be related to what we are trying to calculate. For instance, the pre-H, the H region, the rising H region can be related to the geometric structure of your material and to the metal ligand overlap and some of the properties that are of interest to people who do with chemistry or surface science. And the pre-H region, which is usually this kind of region, I think the previous slide shows it, but the pre-H region is more sensitive to the ligand field, spin state, and central symmetry. But for this presentation, we are more interested in the geometric structure, on which I will show you. So what exactly are we talking about? So we take a case study, for instance. So this is the schematic of the Earth interior, the core of the Earth. So in the core, so there is iron. So what has been postulated is that the core of the Earth is contain iron, and of course iron alloys like iron nickel, iron hydrogen. You have this kind of iron sulfide and several alloys in the Earth core. But the challenge is that over the years, people perform experiments and they say that, okay, let's focus on this particular one, for instance. So the challenge is that people always usually argue that what exactly is the structure of iron at certain pressure and temperature conditions. For instance, if I look at this point, which is about 80 gigapascal, for instance, and about 3,000 Kelvin, iron is liquid. For instance, this is what an experiment that was performed some time ago. But you see some people saying that no, it is not the case. Iron is actually liquid at this point. And then you have all this kind of controversy that has been over the years, and up to today, people still argue about this kind of thing. So in actual fact, what we have here is a kind of an aggregate of the results people have obtained over the years. And what seems to be acceptable, what we consider to be correct, is that at about 78 gigapascal and about 3,000 Kelvin, iron is liquid. But how do you, in the laboratory condition, how do you kind of confirm that? So in 2009, a group in Germany, they did an experiment using X-ray absorption. And what they did was that they basically took iron, HCP iron, and they subjected it to some pressure of about 80 gigapascal, and increased the temperature gradually. And what they told us from their X-ray absorption structure features is that the edge peak. At room temperature, they see this kind of peak. At about 2,300 Kelvin, the peak is like the intensity reduces. And at 2,900 Kelvin, this intensity, you can see it is this one here. Later on, it reduces a bit, and it completely disappears at about 3,000 Kelvin. So they said this is a signature of melting. They said because of this, iron is actually in liquid form. So they use this laboratory experiment, X-ray absorption, to basically confirm what has been done over the years. So when we saw this, we were a little bit we were excited about it because that time we were working on, we were studying classical potential for some metallic system. And then one of us has already, we have this kind of this modified embedded atom potential that somebody was working on for a different purpose. So we decided to repurpose that particular research, and we say, okay, can we actually use this? Can we develop this this classical potential to produce to produce ion structure at 70 gigapascal and 3,000 Kelvin, and then use the structure produced from this classical potential and do the extra absorbance calculation and see whether we are going to observe to calculate exactly what they got in the experiment. So basically, we want to use our own classical potential to produce ion, a liquid form of ion, and then do the X-ray absorption, like what they did experimentally in the laboratory, and see if we are going to get the same thing. Basically, we want to see the fidelity of our classical potential, whether our potential is doing well, whether we are on track. So that's exactly what we did. So we did ab initio calculation. We did force fitting forces and stresses. I don't want to bother you with that. It's a little bit cumbersome. So we generated our classical potential, and then from there, we used that classical potential to generate ion at exactly that. Okay, so one thing about the classical potential we developed was that it was mainly for ion at that pressure and temperature condition. It is not flexible. It's just for that particular pressure and temperature condition. So then we produce ion, room temperature ion at that pressure, 2,000 Kelvin and 900 Kelvin. And then we use this, and then we decided to, then we did the X-ray absorption calculation with that. So what we see here is a comparison between the experiment, like the one I showed you, and our calculated. The calculated ones are the dotted one. So in a way, we fairly reproduce what experiment is the full lines, ours, the calculated one are the dotted lines. But what is important here is that, sorry. So what is important here is that, indeed, at about 2,900 Kelvin at 78 gigapascal, actually, that edge feature, it disappears. And our classical potential and our X-ray absorption calculation agree with what has been determined from the laboratory. So, so that's just the message at this point. So the little discrepancies you can see from here is basically due to the fact that our calculation neglected many body effects. That's the limitation of the theory itself. And because the experimental sample contain grain boundaries and things like that, it's not defect-free, unlike our ideal atomic structure, which we use for the calculation. And so we are excited by this, that okay, we are able to use this X-ray absorption to study structural materials. Then we decided to, so a few years down the line, we decided to move on and do something else with it. So, so we start to consider the cobalt oxide. The interest in this is that somebody was interested. Okay, so there was an experiment where they were trying to use the cobalt oxide to basically study the 2-propanol oxidation to acetone on the surface. So we didn't want to jump to the surface right away. We said, okay, let us look at the box structure as a template to study the surface, in the future. So we build the structure, and then we do the basic electronic structure calculations. We apply U of 3.5, and then, so what happened is that without U, we have this over U. Without U, we have 0.34 as the electronic band gap. With U of 3.5, we have 1.72, which is reasonably, we compare reasonably with the experiment. So with just simple application of U equal to 3.5, we have electronic band gap that is close to the experiment. There's nothing complicated there. It's quite straightforward. So then we move on from there. And then the experimentalists, they did the experiment with X-ray diffraction. They obtained this kind of diffractogram, which they say is actually the cobalt oxide bulk they were trying to synthesize in the laboratory. So then we obtained the X-ray absorption as we normally did. So the experimental spectrum is this particular one here. And when we did our oxygen KH, we have a structure that is fairly consistent with the experiment. Okay, so this is just the spin resolved spectra, which is basically it's not too much different from the one we obtained without any spin polarization. So, so the next thing we did was to kind of do the electronic projected density of state electronic structure calculations to kind of try to understand further where those spectral features are coming from. So if you look up here, you have the oxygen 2p state. If you compare that with our oxygen KH, you see that similarity there. So this basically tells you that spectra we got from our X-ray absorption is due to transition from S to P orbitals. And then, I think the time is up. So what we did thereafter was to kind of determine the electronic configuration of the cobalt atoms in cobalt oxide. And we did some density of state, orbital resolved density of state analysis, and we came up with this kind of crystal splitting diagram, which we have confirmed that it's actually consistent with what is known before. So, and then, so our X-ray absorption allows us to kind of understand the electronic structure of cobalt oxide. And then the next thing the experimentalists want to do is to actually kind of create a surface, and then use this surface to do the kind of catalysis they want to do. And because now we have a good understanding through our X-ray absorption, we can use the X-ray absorption to probe electronic structure. Then hopefully we'll collaborate more in the future and see exactly how the collaboration goes. So the experiment is being conducted in Germany. They are doing the X-ray measurement there. And thereafter we will speak again and see exactly what we can. I think I will stop here. But before I stop, let me just, I want to go this way, but I won't be able to do that. Let me just quickly go to this. Anton is here. Anton, please pay attention. So basically, this is the kind of script we use for our quantum espresso. And I'm asking myself, this particular one, this ABE, I think the meaning is that when you start your calculation, if it aborts, you get an email, right? If it starts, you get an email. And if it ends, you get an email. We don't get any of those. So this doesn't work. Okay. So we have an in-house HPC at UNISA. This works for us. Okay. So you can imagine you submit a job, and then the job has crashed, and then 2 days after or a day after, you get there, you don't get any email notification. So please look into this. The last time I talked about visualization, that we want to visualize our atomic system on the HPC. We don't want to download our data from the HPC to the desktop. We want to, it's a little bit cumbersome way to do it. I don't know whether there's been any development on that. And then I asked one of the students, what are the problems they are facing? One of them said, I want to know if there's any way to reduce the amount of time we spend waiting in the queue for the cluster. Are there any tricks or tips you can share to help me submit my job faster? I would greatly appreciate it. So I simply copied the email and pasted it here. So success, well, we have been able to have like 6 publications per year, thanks to the HPC. We have collaboration, which is African-wide and beyond, because we have some things that, we can do some things that are of interest to the experimentalists, so we talk more to the experimentalists to talk to us. This enhance the quality of our research, so we are grateful to the HPC. So again, our appreciation to the HPC, NRF, through their funding. Then we have 2 postdoc positions which have been advertised on NITEC's website. Thank you very much.
Which questions should we address first, the presenters or the audience? Okay, I'll ask the audience.
Please.
Any questions?
Yes, please. Yes.
Thank you, Abdul. Really, really interesting work, especially the high pressure, high temperature stuff.
Yeah.
As a metallurgist, that's very interesting to me.
Thank you.
I was just more going to ask a technical question. These sort of simulations, I mean, you're obviously getting a lot of questions from your group about how to get them on the cluster faster and everything. How large are these simulations, and how many, what kind of resources do they require when you're running on CHPC?
Okay, so for the classical molecular dynamics simulations, we do this on our desktop because we use classical potential. So with about 250 atoms, I think, that could be done. But when it comes to the S3 absorption, so we have to use the HPC. So we take the atomic structure from the classical molecular dynamics simulations, we run that on the HPC. So that. Okay, so typically what I think I showed something here just now. Usually we use 72 CPU. Where's that now? Aha, okay. Usually we use between 48 and 72 CPU. That is typically what we do. And most of the time it's sufficient. Yes, that's the thing. Yeah. But again, we discovered that sometimes increasing the number of CPU does not really help. So there has to be a kind of balancing act between the,
Good afternoon, everyone. My name is Tawani Ramlongo. I'm a researcher with a virology background. So I'm going to present the work that we are starting within the Indigenous Knowledge System program. So our program falls under the Advanced Agriculture and Food within the agro-processing group in the CSIR. So the presentation, I will give a definition of IKS, what IKS is all about, the NICMAS, the database that we will be using, the WHO priority diseases, RNA viruses and their prevalence, challenges in conventional drug discovery, and the approach that we're going to take for this project, and the prospect for the project. So Indigenous Knowledge System is all about the knowledge, the skills, the philosophies, the practices, the know-hows, the learnings, the technologies, and innovation that the communities has developed over time. And this is in a specific location. So these systems are developed by communities through the interaction with their natural environment to survive before modern technology. So IKS is cross-cutting and multidisciplinary field that finds applications in agriculture, health, food and food product preparations, and environmental conservation. This knowledge has been handed down from generation to generation, and therefore the idea of knowledge ownership and protection is crucial. So in this project, we're going to tap into the indigenous knowledge so that we can select plant species that we're going to use for drug discovery. So for that, since we'll be using the indigenous knowledge, we need to comply with the legal frameworks that are in place in South Africa to protect IKS. First, we have the Biodiversity Act that has been enacted in 2004, along with the regulation on bioprospecting, access, and benefit sharing. Secondly, we have the IKS policy, which affirms the use of IKS as a source of technology and innovation. So recently, the IKS Act, which is the Promotion, Protection, Development, and Management of IKS, which has been enacted in 2019, this act is one of its kind because it protects the knowledge, the indigenous knowledge. So instead of going through the application for IP, intellectual property protection, automatically the knowledge has been protected. So South Africa is not only rich in indigenous knowledge, but also in biodiversity. We have more than 20,000 plant species, in which about 9% of it is used for medicinal purposes. So the DSI initiative, which was developed by the CSIR, produced this platform which is used for management of indigenous knowledge, and this platform links the medicinal plant species to the indigenous communities. The platform is connected to the IKS documentation centers in selected provinces, and then they are there to support communities to record their indigenous knowledge. The information is accessed and published through this system, has ownership completely vested in the communities. And then you cannot just simply access this information, you need permission and authorization. So in the IKS space, for us, we are focusing on respiratory infections. So when you go to the database, you find the information on the plant species and their indigenous uses. So because in the indigenous space, they do not say, okay, let's say like the virus causes flu, so they won't say like this plant is used to treat flu. They will just say like it's for treatment of cough, of blocked nose, something like that. So we use that as guidance to identify the plant in this database. So at the end, we end up with 62 plant species that were used for treatment of respiratory infections. So there's also, I think all of us from the experience of the COVID pandemic, we have stories that we can go on for days. So the WHO has listed diseases that are priority for research and investment. So in this list, the COVID-19 still takes a spot. There's Lassa fever, Rift Valley fever, Meg fever, Ebola virus disease, Nipah virus disease, Zika virus. And there's also another one that made the list. Okay, I think I'm going to spoil the disease X. So the disease X is specifically there because there's always a potential. The experts, they recognize that there's a potential of an outbreak, of next outbreak that can be caused by a pathogen that we have never experienced before. So that's why there's also a spot for disease X. So these diseases, they pose the greatest public health risk because of their infectious potential, with the potential to cause a pandemic. And currently, there's limited countermeasures, and therefore they are prioritized for research and development in public health and emergency context. So like the WHO is encouraging the global research community to invest resources to develop diagnostic tests, treatments, and vaccine. So the one most important thing to notice about all these, also including disease X, all these 9 diseases, that they are caused by RNA viruses. So just to give an overview of the prevalence of RNA viruses in humans in the past 26 years, the biggest challenge with RNA virus is that we have with RNA viruses is that they are zoonotic. So that means they infect both animals and humans. And also the second problem is that when the genome of the RNA virus, so when it multiplies, that there's no proofreading, so that means then there's mistakes. So we know from the COVID-19, there was one variant today and the other variant tomorrow. It's because of that lacking of proofreading. So in the past 26 years, we have seen also that there have been the ones in red. The ones in red, so these ones are the outbreaks that have been caused by the spillage from the animals to humans. And the ones like the SARS-CoV, the MERS-CoV-2, and the SARS-CoV-2, those are the major outbreaks that we have experienced in the past 26 years. So the other problem that we have with these RNA viruses, these viruses that the WHO has identified, is that you can't just handle them in any facility. So you need a biosafety level 3 facility to handle the viruses. And then with the conventional drug discovery, you need the whole pathogen, or you need a protein that you can use to do the screening in the lab. So then with the lack of being able, the facility to handle the pathogen, it makes it almost impossible. So, and then if then you option of using the virulence protein, that means then you're going to have to go through the expression of the protein, all that, which then takes much longer before you can actually start the screening process. So just from the target discovery to the lead compound validation, it will take you about 2 to 3 years if everything goes as planned. And then you still have to go preclinical development and clinical trials, which takes even another 9 years or 10 years. So that's quite long. So for this project, we decided instead of going these routes, down here, which is the conventional route, which is slow before you get to the in vitro testing, we decided to use in silico modeling, which is faster. So because then you just need to use the soft structure of the protein, the target protein that you are focusing on, and then the compounds, and then you do the in silico modeling, which then speed up the process into in vitro testing, and then clinical trials, and then have the drug. So because of the diseases that the WHO has identified, they are caused by RNA viruses, we decided to choose the RNA-dependent RNA polymerase as our target protein. The reason of that is that all the RNA viruses have this protein, and they have the conserved region with the way the RNA interact with the protein. So here is the structure of the RNA-dependent RNA polymerase solved through cryo-electron microscopy. And then if you can see the circle, build like more a circle, this one is your RNA strand. And then there's a dot here where it shows the active site. So this active site is falls within the NSP, which is this here, the NSP12 domain, and then the motif A to E, they're the ones that are conserved. So even though the RNA-dependent RNA polymerase might differ between RNA viruses, but then that region right there is conserved, because that's the region that interact with RNA. You can also see here from, if you turn, then you can see there's your RNA strand, and then the active site is somewhere there, is where it interacts. So, so that means then we will be screening for a drug that will have a broad spectrum. That means it will be able to inhibit across the RNA viruses. So for this work, we're going to start by creating a database of compounds from indigenous plants. So I've mentioned that we have identified the 62 plants from the NICMAS database. So there's also very limited literature on the phytochemicals of the indigenous plants. So some of them we might be lucky and be able to get the information from literature, but some of them we're going to have to do the profiling ourselves to create this database. Then from there we'll we use the Schrodinger method through the CHPC platform to do the docking studies. So we will do the molecular docking on the RNase-dependent RNA polymerase with the phytocompounds. So here is the structure of the SARS-CoV-2 RNA-dependent polymerase. And this one is already being docked due to the, I'm not sure if you can see here, but there's a compound there which is the remdesivir. So the remdesivir is FDA-approved drug that we use for treatment of COVID-19. So we'll be using that one as our standard. So then after the molecular docking, then we'll do the molecular dynamics simulation, determine the stability of the complex, and then followed by admit modeling. So and then when it comes, so this all modeling stuff is still very new to me. So, but then when it comes to the validation of the lead compounds, that's way more, at the moment, I'm more familiar with. So we plan on also developing assay that will be used to validate the lead compounds that will be generated from the model, and then also see if they really inhibit the virus replication through inhibition of the RNase-dependent RNA polymerase. So just as a case study, we chose Kigelia africana. That's one of the plants that we have identified from the NICMAS database. This plant is generally known as a sausage or cucumber tree, and then it's found in Gauteng, KZN, Limpopo, and Mpumalanga. So this plant is traditionally used to treat cell anemia, epilepsy, respiratory, and digestive problems and so on. So like in literature, there's extensive information on the compounds, on the profile of the compounds of this plant. So that's why we decided to use this one as a case study. So there is 152 compounds that have been identified, and then we did the molecular docking on the SARS-CoV-2 RNase-dependent RNA polymerase, and then of these compounds. So we did protein prep, liquid docking in Schrodinger software. So out of the 152 compounds, also including the conformers that were generated when we did the liquid prep, we found, and then when we did the docking, the molecular docking, we found that there were 2 compounds that shows like very good scores. You can see here the scores, the scores for remdesivir, which is -10, and then for compound 1 and for compound 2 was 11, around -11.1, and for compound 2 -10.4. So I'm not going to try to explain the figure. Hopefully I will do that for you in the next conference once I am more comfortable with this data. So for this, we'll still do validations, which includes molecular dynamic simulation, ADMET analyzing, and also the confirmation on cytotoxicity and viral inhibition. So where we're going with this project, we want to have a comprehensive online database of active phytocompounds linked to plant species, which will also have their GPS coordinates, traditional use, communities that are linked to the knowledge, the biological properties, and the clinical data. So we will be utilizing the CHPC platform to process, because we plan that this data is going to still grow, because like for only one plant we had around 152 plants, and then we still have to have the list of compounds for the other 61 plants. So then we will then be able to utilize the resources then of the CHPC. So where we're going, also our aim is to fast track the indigenous knowledge to value-based product development, And this means that then the communities will benefit through this development, since we will also engage them on benefit sharing agreement. So we will develop an in-house in vitro validation of lead compounds, and also we already have the capabilities of product development, which means that this process will be established. In the case of the next pandemic, then we will have the process from the beginning to end in an accelerated manner. Thank you.
Good afternoon, everyone. A very interesting talk. However, can I ask one thing? You started doing molecular docking, and then you are going to do molecular dynamic simulations before you are going to screen for ADME properties. Or you started with screening or profiling the ADME properties of the compounds for your drug discovery process.
So yes, we started with the molecular docking so that we can have a reduce the number of the compounds that we will put on ADMET. So that we will,
okay.
Yeah.
So I understand that. Don't you think it's more feasible for you to start profiling the ADME properties, which are your drug likeness properties of these compounds, before you can actually conduct molecular docking and molecular dynamic simulations? Because what if you consider all these compounds for molecular docking and they give you good scores? However, when you are done running your molecular dynamic simulation, you found that some of these compounds are not drug-like molecules.
So that means that those compounds will fall off.
You see, so instead of just going to the HPC cluster and running a lot of molecular dynamic simulations for compounds that will not be relevant for drug discovery process, why wouldn't you start with these ADME properties?
Krishna, do you want to help me?
Relevant, but the first question is, I know some of the pharmacists use this for medication. They use this indigenous knowledge, and they use some of these plants for medication, and of course, I'm sure it works. Then the question is, how does the Medical Research Council treat this kind of information when it comes to patenting? Do they accept it, your studies, or what do they do with it? Do they, I mean, of course, there is Western type of medication that we use, of course, but this one, if it's a medication, do they accept it? Do they patent it? What happened with it? That's what I want to know.
As long as there's scientific data that backs the claim, then yeah, it is possible to register the drug with SAPRA, and then you can be able to find it on the shelf. So the most important thing is that to have data, to have clinical data to prove what you're saying. Is it safe? So that, yeah.